the base, right, that this is closed, and then open it 2 turns. Right, everything is nice and tight. And turn on the gas. Make sure I generate a spark. Got too much something. There we go. Too much gas. Turn that baby down. Am I going the right way? Yeah. Okay. There we go. There's 2 arches. Done with my flame. So I have my loop. I've put a paper towel so you can see, right? And you'll see I've labeled my slides 1 EB for my initials, 2 EB, 3 EB, 4 EB, which goes with my sheet, right? Later you can use these little labels after you stain and write a more descriptive label if you want, and a more permanent label on your slide. Part of the reason is for identification, also to know which side is the top of your slide. So I'm going to go ahead and put my gloves on, because I'm going to be working with dyes and I don't want to be multicolored today. And my apron on and my safety glasses. I have all my materials from my material list, and some of them, I would be sharing with my lab partner. I have an additional slide you'll see that I didn't label. That's my spreader slide. So the first 2 slides I'm going to set up, and you can set them up together, is my negative staining. So I'm using nigrazine, which is an acidic dye, negatively charged. I put a drop of dye close to my label on each slide. Then I'm going to aseptically transfer bacteria, so I'm going to sterilize my loop to handle, flame it orange, kill anything on it, sterile loop. Now I'm going to go, the first sample I'm taking is from my Petri dish. Remember, I want to prevent airborne contamination, so I want to keep my lid over it and just sneak in from the side. And I have 1 colony right there. Who knows, maybe it's an isolated one. I'm going to pick it up. I'm going to add it to this drop of dye, just mixing it in, not necessarily spreading out the dye, just mixing it in. Now you may pick up dye, like sometimes you'll end up with the whole loop full, right? Try and knock it out, or don't worry about it because you'll flame it away. It's just going to take you longer to flame that excess dye off. So I'm going to flame it away. And now my next one says, what sample am I going to do? You, my lab partners, help me out. I'm going to do Klebsiella, and it's a slant, right? So I want to angle it so I can see. You can see I got nice growth. Right? Grab my cap, flame the mouth, and again, just a dab, right? Flame, cap, put it back, and then introduce the bacteria into the dye, mixing it in. Flame to sterilize my loop. Now this is 2 different slides, 2 different organisms, right? Maybe. We'll find out. We'll look at it under the microscope. So I'm going to take my spreader slide, I'm going to hold at the label with 1 finger, take my spreader slide at a 45-degree angle, spread out that droplet, push it forward. It doesn't have to be perfect, you're just spreading it out. That's just the purpose, is spreading it out. I already used that side, so to save, right, I'm going to flip it over and use the other side. And just spread it out. Immediately when you're done, put it on your light box, because this needs to completely dry before you can look at it in the microscope. These 2 slides, done. Till they're dry, then I can look at them under the microscope. These guys are a little bit more complicated. We're going to make bacterial smears. So for 3, I'm taking from a Broth Culture, which you guys saw me mix up earlier, so it's nice and suspended. I'll flame my loop again. And because this is a broth and it's liquid, it's going to allow me to be able to spread it out on the slide and create what we call a smear. So I'm going to go in, mix up and pick up, put a drop on my slide, flame, cap, put it back in my rack, and then I want to go from the center and I want to circle out, spreading it out as much as I can over the entire surface. Now I'm going to put it on my light box to completely air dry. When I'm done, it should look like a dirty fingerprint. that would mean that I have a sufficient amount of bacteria, but not too much. Too much bacteria, they're going to be stacked on top of each other, and you won't be able to see individual cells, which is the purpose of looking at them, right, is to see the individual cells. A broth culture usually is pretty easy to make a smear from. A solid culture, on the other hand, we have just bacteria, right, on the surface. So in order to spread them out, we need to add a drop of water. So I have little droppers, right, for you. This is just distilled water. Put one drop in the middle of your slide. Sterilize my loop. And again, just a little dab of bacteria, because I don't want too much. And so this one's going to be our E. coli, right? From the slants. So there's my E. coli slants. Got nice growth on there. Flame, go in. Remember, just a dab. Right? Barely a little speck taken. You don't need to scrape the whole surface and have a big white mess on you. Right. Alright. Of course, I don't even remember which side. Doesn't matter. Put it in the water. Spread it out. And you can see it's a little white. You want to spread it out as much as you can. Flame my loop because I don't want to contaminate my environment. What do I do next?
Air dry keeps it.
Right, so it's got to air dry. So speed that up. I'll put it on here. And you can see these two are almost already dry, except for where there's some extra pools on there. Another second for this one. Okay, so now I'm going to get my lab bench ready for the next part, which is going to be to heat fix these two slides once they're completely dry, which I have 0 patience for. One of them will be dry in a second. When you go to look at your slides, you want to look at your negative stain slides first. These are easiest to focus because you have all this background to look at. What magnification do we look at first? 10x, which is low power. Total magnification of how much? 100. That's like nothing with bacteria, right? What do we got to go to actually really see something next?
Oil immersion.
Oil immersion, right? Focus at low, put your oil on your slide, go to oil immersion. Remember that oil is going to fill that gap in between. If it's not filling that gap, you're not in focus. Which adjustment knob?
Fine.
Fine focus, right? It should pop into focus. You shouldn't have to turn it a lot. Okay? You should, when you look at it, it should be a grayish film under low, and then at high, right, you should, at oil immersion, excuse me, the highest magnification we go, you should see the cells clear, right? And the background is going to be crappy. Now you're going to have other junk on your slide, like dust and stuff like that, but you guys already know, what are we looking for the bacteria? That is shape. What are the two shapes that we're going to see all semester long? There's going to be different colors. Cocci and bacillus. So if it's E. coli, according to your sheet, what are you going to see? Rods. If it's Klebsiella pneumoniae, rods. If it's Staphylococcus epidermis, smears in clusters. Right? Almost there. Which should be our slide 3. Right? Dry. Okay. You can use the clothespin for flaming so you don't accidentally flame yourself. Some students, as you can see, have used it for staining. It's not for staining. Right? It's flaming. One little section, just want to dry. I want you guys to see. This one is just about dry. This one isn't quite dry yet. But do you see how it's just a smudge? Like a dirty fingerprint? You all see that? You're going to have to tip your body so you can get at an angle. These are going to be awesome. Exciting. Okay. So this one's dry, this one's still got to dry, so I'm going to leave it there. Right? And sometimes I just don't use because I know where the flame is, but you know, for beginners, use the clothespin. 1, 2, 3, 4, 5, that smells good. Yep, it's hot. It's done. Right? Put it on your staining tray, you know you heat fixed it. Right? You know you're done. Okay, so what does it say for dye for this one? 3? Yeah. Methylene blue. This is my favorite color. Just flood the whole slide. I'm looking at my clock the second hand. How long am I doing it for? The good old golden rule of a minute. When the minute's up, what am I going to do? Rinse with distilled water, right? These caps, loosely opened, will actually squirt. A minute lasts forever when you're watching the clock. The second you walk away, a minute is up. Right? So we have the clock there, the clock there. We have little timers on the desktop. I wouldn't necessarily use your expensive cell phone unless you have, you know, the life-proof case like I do. Okay, you got gloves on, right? You don't need this. You're just going to fumble things around and break your slides. Just use your gloves. You pick it up and you rinse it off. Then, one slide, right, at a time. Make sure your surface area is flat, because if it's not and you press, what happens to your slide? You break it, and then you have to start all over again, and you won't be happy. You just put it in your book, right? This is just filter paper, right? It's not going to get it completely dry, but dry enough. But you can see I got a nice purple smudge on there, or blue, actually. It's purplish, but it's blue, right? There might be 2 overlap there. I'm going to have to go to the edges probably to see individual cells under the microscope, right? But this slide is ready for viewing under the microscope. Okay? And as soon as this one finishes drying, I'm going to heat fix it, and I'm going to stain it with what? For how long?
1 minute.
1 minute. Then I'm going to rinse, blot dry, right? And then you're just going to go down your list and look at them under the microscope. And hopefully we see what we're supposed to see for each one of the species, right? And we're going to draw and write what we draw and be very descriptive, answer all the questions. If you really like color, I even have colored pencils I can take out for you today. Okay? Go have fun.